Hi, my name is Yari and I was given the KRAS protein for my project. One of the normal functions of this protein consists of giving information from the genes to make a protein called KRAS, and it's a part of signaling a pathway. The pathway tells the cell to grow or split to take on a different function. KRAS protein is known as a GTPase. It converts GTP molecules into GDP molecules. The KRAS protein doesn't work when GTP is being converted into GDP. When KRAS is converted to the GDP protein or connected to the GDP protein, it doesn't give signals to the nucleus of the cell. I picked this category because I wanted to know more information about what goes on in the cells of cancer and how it is started and how it can reproduce. This cell is mainly found in cancer genes or on the plasma membrane of a cell. It can be found in the lung, colorectal, and pancreatic cancer of the cells of the cancer. The organisms that can contain this protein consist of Homo sapiens or humans. This protein can become less active when it's converted from GTP to GDP, and it doesn't give signals to the cells of the nucleus. The protein can become active when it grows and form different cancers. It can go from active to inactive quickly. Cell transformation begins to increase, and then it can become resistant to treatment. The protein gains a new function when the protein will switch the course of sending signals. The change in structure can impact the mutated protein's function and can cause malfunction in the protein. The functional groups consist of GDP, GTP, homeostasis, and glycine. The KRAS protein is hydrophobic, and I would characterize its size to be a little bigger than the other amino acids. The original amino acid side can participate in transforming glycine to valine. The glycine side is smaller, which makes the other side of valine, which is bigger, to stop the binding of the protein. When mutations occur, the amino acid side becomes bigger. The functional groups consist of codon, glycine, GDP, GTP. The mutated side chain is hydrophilic, and the protein is smaller than the original. The mutated amino acid side can form bonds that are stable, so the protein won't grow rapidly. They can consist of cysteine, covalent, and sometimes hydrogen bonds. When a mutation occurs in the protein doesn't fully function correctly. When you are changing the order of the protein, it goes through mutation. Certain instructions of the protein can be missing when it goes through mutation. When mutation happens in the cell, sequences of this protein can become damaged. Mutation can affect protein by changing the shape or size. It can also cause death in the cell. So the first protein I'm going to show you is the original protein. So this original model of the protein consists of the GTP loop and switch 1 and switch 2. So in the switches, the GTP loop gets bonded together with other molecules, and it can form a loop around it and a P loop. So the switch 1 and switch 2 keeps both of it together. In this video, you can see the mutated gene of the protein. So during the mutation, the GDP gets attached to the KRAS gene, and it goes to GDP. And then the information from the GDP goes to GTP. And then that information goes to the KRAS protein again, and the GTP protein gets attached to the KRAS protein, which goes to an active KRAS protein. And the signaling can go down when it comes to mutation. So you can see the loop going around it to cause a new mutation. You can see the two sides, the purple ones, and then the red and the green is the GDP and GTP and the protein together. Now you are looking at the original protein model and the mutated protein model. Some of the limitations can consist of you can't see the signals being sent from the nucleus to the protein. So that's one limitation in the model. But now you can see what happens, the difference between the GTP and GDP, and the difference in the GTP and GDP in the mutated one is more GTP and GDP information in the mutated than in the original. Another limitation in the project consists of not being able to see the amino acid side switching from bigger to small in the project. If I did it online in another 3D model, you will be able to see it. You can see it grow and shrink. Also, you can see the physical difference in the protein model, and that can be another limitation.